Hello biologists. I am just making a short video to talk you through the nucleic acids, proteins and gene regulation test that you have done. Hopefully this will help you to build your error log and therefore close any gaps in your learning, ensure that you don't make the same mistakes next time, and offer you an opportunity to see where you're still stuck and come and find me, or Miss Kemp, of course. I'm just going to run through section B rather than multi-choice because that should be fairly self-explanatory. Most students did very well in that section. Looking at section B, first question was straight from your workbook that you used in class. This is this one here. Okay, page 18 in that. If you've completed that and you've revised it, then hopefully you had some examples of proteins that you could use. If not, you need to go back and do that. Also, as you move through Unit 3, there will be many examples of proteins that you will discuss in different contexts, and you'll be able to complete that table in great detail later. The main thing I want to identify, though, is that, firstly, the example of enzymes was actually in the next question. Rycin's an enzyme, and it states that in the next question. So you can always use examples from that paper. Many people said that enzymes break things down. It's not necessarily true. So enzymes that catalyze anabolic reactions actually help to build things, more complex molecules. So that wouldn't have got a mark at all. You need to say that they speed up or catalyze biochemical reactions. The hormone one, this was a flashback to year 9. Hormones are chemical signals that are released from endocrine glands, and that's really important terminology, but we'll do more on that later. You wouldn't have got a mark if you had given the example of testosterone or estrogen, because neither of those are actually proteins. The monomers are not amino acids, they're cholesterol-based hormones. So it's just good to note that some hormones are protein-based and some are not. I've given you some examples here of ones you could have used. Now, if you weren't clear on what the proteome is, go back and watch the video links about, or even do a Google search, and there are lots of TED Talks and all sorts of resources available on the internet, because proteomics is a huge field that's growing all the time. It's really important to answer this question, section B, by explaining the proteome as analysis of all the proteins produced by an organism, yes, but it could also be the proteins produced by a tissue type or a group of cells, or by a single cell. We can analyze the proteins produced by a cell or a couple of cells. And they're huge areas of research that you can't overlook in this definition. Applications include medicine, drug development, biotechnology in general, but you couldn't just say it was interesting. Scientists like this stuff because there needs to be a purpose in science. Okay, you need to have an objective with a really meaningful application and outcome. So you need to have a think about how you approach those questions and be really specific. Question 2. This is one I really wanted you to annotate, and I noticed that in this test, because I didn't give marks for annotations, many of you didn't annotate at all. It needs to be a habit, and I will give you many more marks on topic tests to help get you in that habit, but it needs to be just something that you do, because it's more active reading the question. You're less likely to miss an important word or section or meaning in the question if you've written on it. Okay. Here I wanted you to really think about what ricin is. Ricin's a protein, it's an enzyme, and it's a toxin. It's built of many amino acids, look, it tells you that. And it's 2 polypeptide chains, and it shows you the structure here in what we call a ribbon diagram. A was direct recall. If you're not clear on your protein structure, go back and review that. It should have been 4 easy marks for you. Down to section 4 here, you needed to say that a sequence change would have led to a different polypeptide chain, which would impact the 3-dimensional tertiary structure of that protein. Therefore, potentially change the function of it They need to be really specific, so therefore reducing ricine's ability to be a toxin. Okay, so making ricin less toxic. That shows the examiner, or me, that you know that we're talking about ricin, and you know what its function is, and you know that it will become less toxic if there's a change. It implies up here that its ability to perform its function depends on a number of specific things. So in all likelihood, any changes would reduce its toxicity. So they're the words I want you to use. Always refer back and use the words from the question to keep your answer very specific and not a general answer. Biology, particularly Unit 3, you must be very, very specific. Here, ricin is classified as ribosome inactivating protein. Okay, annotate that section. Describe how inactivating ribosomes in cells will affect the cell. So you know that ribosomes are the site of translation. Therefore, inactivating or turning them off will mean that translation can't occur and protein synthesis cannot occur. Why are proteins important to the cell? That's how you could have got your second mark. Ribosomes. Why are proteins important? Because proteins, many enzymes are proteins, or most enzymes are proteins, and if you don't have those proteins, you can't catalyze the biochemical reactions, and most functions of a cell are reactions that involve proteins. Okay, so the answer we were looking for is, for 1 mark, tell me that without a ribosome you wouldn't be producing proteins, and then the importance of proteins in biochemical processes. Many people didn't get both marks for that one. You might have got 1 but not 2, so show me that you can get both. Question 3, pre-mRNA, most people got that one. Question 4, this is reading the codon table. Remember that these are messenger RNA codons listed, and the amino acids are the words. I wanted to see you working out, and I want to see it in all of your SACs and all of your tests so that I can follow your thinking. You need to convert DNA to messenger RNA and then read the table and identify which amino acid is coded for by this DNA triplet. Okay, DNA is not codons, it's triplets. That one was mostly fine. Here. Okay. Question 5. Urgently needed annotating. The diagram below illustrates the steps involved that are involved if transcription occurs. Process X produces a copy of the DNA molecule, and this is the important bit that most people missed, and modifies the product. Okay, so X describes not only producing the pre-mRNA, but then modifying it and allowing it to exit the nucleus. If you see that part there, therefore you couldn't just describe the production of a pre-mRNA molecule. You needed to talk about the capping and the splicing. The terms we were looking for: RNA polymerase, the promoter region, elongation by reading the template strand, or the enzyme reading the template strand, adding free nucleotides, ideally to the 3' end, to build the pre-mRNA molecule. This process stops at the termination region where RNA polymerase will detach, and messenger RNA then must be processed before it can leave the nucleus, which involves the removal of all introns, some exons in the case of alternative splicing, but that was additional information, you didn't need that bit, and the addition of a poly A tail and a methyl cap. This part people struggled with. What was the advantage to being able to switch genes on and off? The advantage is to conserve energy and resources. Energy wasn't enough. Not even energy and resources was enough, because it's a 2 mark question. Okay, so energy and resource conservation would be 1 mark. How does it conserve those resources and that energy? By only producing proteins when needed. That's your 2nd mark. Okay, conservation of energy and resources by only producing proteins when needed. Look at the marks and consider what you need to say, how you need to meet the criteria for that question itself. Okay, gene regulation. Describe the events that lead to the prevention, annotate, of the production of enzymes that break down lactose in E. coli. E. coli is the organism. Lactose is the molecule that actually regulates gene expression. So the first step is to describe the fact that enzymes will not be produced in the absence of lactose. So absence of lactose is mark 1. If there is no lactose, the repressor protein will bind to the operator region of the operon, mark 2. Therefore, RNA polymerase is prevented from transcribing the structural genes. Really specific language, please. Therefore, gene expression is prevented and the enzymes are not produced. That's your 4th mark. Okay, so you need to tie it up. You need to link it back to the question. Okay, so that last mark describes why all of that means that enzymes won't be produced. Last couple of marks here. Direct recall, I hope. This one, there's a definite formula to answering these questions. So identify the difference between structural genes and regulatory genes. It's much easier to start with regulatory genes. Regulatory genes code for the production of proteins that are involved in regulating gene expression. Direct memory mark there, so pull that from your mind, write it down, and then explain why structural genes are different. So structural genes or gene expression results in the production of proteins that are not involved in controlling gene expression themselves. So any other protein, but a protein not involved in gene expression. Define the following terms. A repressor protein. This is a protein that binds to an operator region and prevents gene expression. Okay, and the operator. Operator is an example of a regulatory sequence. So it's not a gene, it doesn't code for a protein. It's the region where the repressor protein will bind, and therefore the gene that is kind of the location for that switch. But the words we were looking for was regulatory sequence, not necessary, but maybe a region where the repressor protein binds in an operon. Okay. I hope that makes sense. If it doesn't, please come and see me and make a detailed log of any mistakes or dropped marks or half marks so that you don't do that again.